All right. Hello, everybody. We are going to talk about cell cycle checkpoints today. So this question that I have on the screen, this sort of warm-up question, is designed to test and see if you understand the cell cycle enough to talk about checkpoints in the cell cycle. So if you can answer this question, then you are in a good spot to be able to understand the rest of today's lesson. So take a quick second to pause the video and see if you can answer this question. All right, so this question is asking about the amount of DNA during M phase and how that amount of DNA changes. The correct answer for this question was C. During M phase, the amount of DNA is halved as the cell divides into two daughter cells. So if you didn't understand this question or you didn't get that answer, I encourage you to go back and review mitosis and the cell cycle. But if you got that right, then you should be free to move on. And so we're going to talk today about how this process of the cell cycle, how it is regulated. In the previous lessons, we have learned about ulcers. And so let's say that you are recovering from an ulcer and you are at the end of your stages of recovery. So the cells in that pocket of dead cells, they have rejuvenated, they have been replaced. And now we need to stop cell division. So what is going to keep those cells from continuing to divide? Anytime a cell doesn't stop dividing, it's going to potentially cause cancer. Now, in order to stop the cell from dividing, we actually have internal and external mechanisms that can trigger different behaviors in those cells that are going through cell division, right? So let's use the ulcer, for example. Those cells, once they start to realize, oh, I have other cells around me, and I think we're at a place where we've replaced all of the cells that have died, I should probably stop dividing. And there are sensors and mechanisms in place specifically for that. So first thing that we're going to talk about is the external cell signal. So what are the things coming from outside of the cell, triggering something within the cell that is going to cause the cell cycle to stop? Those three things are growth factors, density dependent or contact inhibition. It's also called contact inhibition, or anchorage dependence. These are all things that can limit the amount that a cell can reproduce. And so I have a video here, and I'm going to just mute the sound on this video because it talks and goes through what normal cell division looks like versus what cancerous cell division looks like. So you can see the phases of the cell cycle here on the bottom. This is just a quick reminder. And those phases are occurring, at a relatively normal rate in this case. There's nothing abnormal about this process of cell division. But then just a quick reminder, in cancerous cell division, if we skip forward in this video just a bit, cancerous cell division is going to occur much faster and it's going to be less regulated. So normally there are pauses in between the phases of the cell cycle, and those pauses do not occur in cancerous cells, and that's what causes these tumors to be able to form. All right, so the first thing that we're going to talk about is growth factors. So growth factors are going to be very familiar to you if you've studied signal transduction pathways. Growth factors are going to bind to receptor proteins on the plasma membrane, and they're going to trigger an internal signal transduction cascade. Now, this video is available to you. I will link it in the description below. I'm not going to play it right now, but this is just a reminder of signal transduction pathways, and I'll show you some of the screenshots of it. This process of a growth factor signaling is basically a ligand binding to a receptor. We've seen ligands and we've seen receptors very, very frequently in this unit 4 of AP Bio. So if we're looking at this cell, and in this cell, we've got a bunch of different proteins. We have receptor proteins, we have signaling proteins that are going to show up, but this is what happens sort of normally, right? So this is your signal receptor. And then as the signal, the ligand, binds to the receptor, this is going to cause that receptor to change shape and initiate a signal transduction pathway. That's normally what happens. In cancer, or in cells that have been somehow mutated to not do this process appropriately, you can get what is called autocrine signaling. So autocrine signaling is when a cell can make its own growth factor. That growth factor is then going to be released from the cell and triggers its own receptors. So this is not a mutation in the receptors in this case. This is a mutation in the transcription factors. It's a mutation in the proteins that make the signaling molecules. So these signaling molecules that the cell releases from its own membrane, there's a mutation in the process that makes these growth factors. And so instead of releasing these growth factors in appropriate times, these growth factors are released and they're used to trigger more cell division, right? There's further evidence that cancer cells have increased gene amplification of receptor proteins for growth factors. So during the process of a cancer cell growth, it's actually going to multiply the amount of receptors. It's going to transcribe more receptors. And those increased amount of receptors mean that the cell is going to be much more sensitive to any growth signals or ligands that will cause the cell to go through cell division. And so this can end up causing cancer as well when there is a mutation that causes an increase in the number of receptors. This is overexpression, gene expression, overexpression of the receptor proteins. So that's what a growth factor does. Growth factors, you guys know pretty well. Because you recognize them in ligands, right? They are the same process that's involved in a regular signal transduction pathway, right? This is specifically showing a signal transduction pathway in cancer, but the ligand is going to bind to the receptor. The receptor is going to cause a conformational change. This is going to translate a signal into the cell from outside of the cell, and that's going to cause a change in the internal cell processes. So this is going to cause a signal transduction pathway. And that signal transduction pathway will eventually lead to the transcription of a variety of different genes that will cause cell proliferation, right? So during cancer, growth factors can be mutated in a variety of different ways. You can have a mutation that causes more growth factors to be transcribed. You can have a mutation that causes more receptors to be transcribed. And we're going to look at other mechanisms that can potentially cause this as well. So we've seen this diagram before as well. I just wanted to remind you, you don't need to rewrite this down, but this is just another way of looking at the same information that we just saw in the video, right? Growth factors are going to trigger things that happen in the nucleus as well as in the cytoplasm, namely transcription and translation. And then those things are going to cause cell division and cell growth. I'm going to move this guy over here. Right? Okay, so we've seen these images before. That one is fairly straightforward. Now we're going to talk about contact inhibition. So contact inhibition prevents mitosis when the cells are in direct contact with one another. So a cell basically reaches out to the cells around it. It's feeling around. It's kind of feeling around blind, and it has to use its receptors sort of like a walking stick to say, oh, there's another cell next to me. I'm going to stop dividing because we have reached our threshold. We have reached the amount of cells that we need, and we don't need any more. Sometimes the cell overshoots a little bit, and that's when you can get things like scars, right? Scar tissue is the increased cell proliferation. And then the cell has to kind of figure out, oh, okay, we're done. We don't need to divide anymore. Now, the way that it does this is through a protein called the CAM protein. And these proteins are going to cause the inhibition of cell division, right? So let's take a look at this diagram. When a cell is going through cell division, growth factors are going to trigger this R protein, and those R proteins are going to trigger subsequent signaling proteins that associate with CAM. And then CAM, because it's not sensing any other cells around it, it's going to associate with this RAS protein, and that's going to cause continued cell division. Okay, this is what happens when the cell is undergoing cell division. Now, in the case that the cell experiences the sensation of another cell next to it, so it associates with another CAM protein, what this does is that this recruits an M factor. Okay, so if we see this M factor in the image to the right, that M factor is going to inhibit the other proteins from associating with CAM. Because the M factor inhibits the other proteins from associating with CAM, it's going to keep this RAS protein from associating with CAM. And that is going to cause cell division to stop, right? So if this M protein was mutated, what would happen why would this cause cancer? This is my question for you. I want you to pause the video and take a second to think about why this might cause cancer.